Okay, now we are going to start the part 4, discussing about more advanced ideas, how these different layers of information is being processed by different maps that we have in cortical areas. As I discussed, we have V1, V2, V3, V4 inferior temporal gyrus, and these neurons are interestingly connected to each other in different ways and different formats. So they have what we call feed-forward information. So we have the information that is going from here to there. It's called feed-forward information. And also we have information that is propagated to the transfer, so the information that is kind of being distributed across the layers. And then there are some levels of information that are going backwards. So it's something like when you realize, okay, this is a sponge, this is a syringe. So when you are a drug user and you see a syringe and you record, okay, now I can see and I can identify this object in my visual cortex. It is a syringe, it is something important. Then it would give information back to the other parts of the visual cortex. They need to have more information from this part of the visual field. So it gives information back to other pieces and facilitate to have more information from the part that you like to see more. So other than feed-forward information, we have feedback to be able to resolve the complexities that we have in other levels, or to have more information about things that we are more interested to attend or have more information about. So that's an interesting part of the system, that it is not just feed-forward. It would have also a feedback system. And that is one of the main reasons that we have activations in other layers of visual cortex. When we show people two images that are exactly the same in terms of the lines, color, everything. But when one image has more information in terms of, let's say, salient information, that there are syringes and drugs and things that are important for a drug user, then the system would start to ask the visual cortex to give more information. So then the visual cortex would start to get more activated to receive more information. So they have done the kind of the basic processes at the same in the first look, but over a time they would get more activated on the side that we have more information. There are information that we are more interested to know about, and that's the reason that they go to the other kind of part of the visual system to be able to extract the information. And then you have activation on those areas. So that is one of the reasons that I told you those kind of maps of brain activations drug users, why we have activation in the visual cortex more than what we have for neutral images, while we know that they are controlled for basic visual information processing. This is because one kind of piece is because of the feedback information that we have in the system. To just give you a better sense how we kind of detect how these areas are involved in the visual processing, for example, this one is a kitty, small cat, you can see. When we show the kitty, the kitten, I should say kitten, yeah. The kitten or the small cat, the visual system, or when we scramble the photo, if the photo is scrambled, the response in the visual cortex is still the same. So the visual cortex is not about object, it's about lines, contrast, illumination, about those info. And this is the same for all these 5 kind of levels of the stimuli that we have. But when we talk about lateral occipital cortex is not very much interested to number 5, because there is no object, because lateral occipital cortex is about object. So if there is no object here, there is no activation in lateral occipital cortex. But we still have activation in the V1. So that is how we know these areas are involved in, let's say, object recognition, not in the basic visual processing. Okay? And there are kind of explanations why we have, when we see these kind of visual illusions. For example, when we see this, we do not feel that this is the same level of illumination in the middle kind of line. Or sometimes we just feel these parts are a little bit blue, or these parts are a little bit red. So you feel that you like to kind of feel that, okay, these areas are not as white as the areas here. And these are the kind of the things that are happening in these visual kind of processing levels. In these visual processing levels, they start to fill the gaps. They start to connect the dots together. They start to seek for something that they were looking for before. So it is not exactly the information that we had from before. It just kind of give you more information, even more than what you are receiving from the environment. Okay? And there are some other illuminations. And this is exactly what we see. What we see here, even these pieces are totally separate pieces, but what you see here, you see a football ball, or what we say here in US, a soccer ball. So you can easily see something like a gestalt of those pieces of information that now, when they are put together, they have a meaning that didn't have that meaning before. So you start to make an object from really simple visual stimuli. And that is how these kind of advanced processing networks are working to make complex concepts from these simple stimuli. It's also important for us to know that sometimes when we see the environment around us, we just feel that what we have around us has the same level of resolution. But in reality, what we see just in the foveal, in the center of your visual system, is really in high resolution. Information from around you is basically, they are not in the details that you have around you are not in the really high resolution. But what you do, you have your eye which is moving and scanning the environment around you. And because you are scanning your environment around you, so you start to reconstruct the environment in the same resolution. So you do not feel that, okay, I have different levels of resolution in different parts of the visual system. You just feel that, okay, the entire visual system has the same level of resolution. So you do not feel that, okay, I just feel things in a better resolution in the middle. So that's not the sense that you have from the periphery and the visual processing that is happening in the environment. And then these visual hallucinations, visual illusions that I'm sure you have seen those before. For example, when we compare these two pieces from each other, we just feel that this one is brighter than this one. But in reality, when we just make a line between them, you can see that they are exactly the same brightness. Why we have this feeling that this one is brighter than this one, but here we can see that they are not real. And even if you just crop them, you do the cropping in Photoshop or something like that. Sometimes it's really hard to believe even that this one has the same color with this one. But in reality, that is the reality. There is no difference between them. This is what you see is just the reconstruction of what is happening outside. And it is true for some other visual illusions, like things that we have. We call them multi-stable or biastable figures. By biastable figure, it means that we have two shapes that are happening here. Sometimes you see faces, sometimes you see just the vase or the glass that you see here, or the table that you see in between. Or even this one would have two different, let's say, geographical shape, whether this kind of side is front or this side is back. So you can have two different shapes at the same time. And sometimes you feel that you are seeing one shape, and the other kind of seconds you feel that you have another shape. These things are kind of giving us a better sense how this system is going to work. So I'm going to give you more details about how these things are being processed inside the brain, and other kind of complex pictures to process. Okay. This is the last slide from this section. I'm going to kind of give you more details about them in the next section, how these things are being processed. One of the other things that is going to be important for us, is going to be critically important. Just take a look inside this picture. It's going to be really interesting. When you give people images without masking, or you give people really sharp images. Let's say you give them pictures for, let's say, 500 milliseconds, or even 2000 milliseconds, something like 2 seconds. So people have enough time to see them. When you give them enough time, they consciously see what is happening. But when you give them lower amount of time, something like 50 milliseconds, they just see something, but they cannot tell you what they are seeing. We call that subliminal presentation. Or sometimes we just make blurry mask over the picture that is really hard to diagnose what is happening under that subliminal presentation of the image, okay? And these colors are representing those presentations. For early visual areas, it is not important whether you're presenting something in conscious or subliminal, without masking or with masking, really rapid, really kind of slow, or really rapid. They have the same activations. But when you go to other areas that are more towards your conscious processing of what is happening in the environment, they have lower and lower activations when you present things in the subliminal order. And it's amazing that some parts in the frontal cortex, they do not even get activated. And that's an interesting part of this visual processing. So when you have subliminal processing, sometimes they do not go to the prefrontal areas. But it might be interesting that for some of these subliminal processing, if I show you drug-related picture, or for kind of those who are not drug users, let's say if I show you pornographic images or kind of sexual provocative images, if I show you those pictures in just 15 milliseconds, they just activate those areas, not very much in the prefrontal cortex, but because of those pathways in the subcortical areas, you still have activations in amygdala and ventral kind of striatum. So we can make unconscious activations with subliminal processes in the basal ganglia and amygdala with subliminal presentations, but those processes are not going to be conscious. And this is amazing how we can activate subconscious processes with subliminal presentations without getting involved those areas that are involved in the conscious processing of visual stimuli. And that's another beauty of the visual system. And that gives me also a kind of a pause or an opportunity to discuss about what William James talked about in terms of what we really perceive from the environment is not exactly what we are receiving from the environment. And it seems that there are many things that are being added in the processes that we have discussed about today to the information that we are receiving from the environment. Based on what I discussed in the first session, if you remember, we discussed about Bayesian information processing. We have a priori expectations. And based on the a priori expectations, what we perceive is not the exact reality. We just update the expectations that we have and see what we are seeing in combination with the expectations that we have and make something new from what is happening in the outside environment. It's amazing that a person was thinking about the same kind of issue something like more than 100 years ago. It's amazing that he's such a kind of genius guy. I really like what he has done, and I will discuss about some of his works in the next sessions. Okay, so that is the end of this part.